Welcome back to our parallel research session. Our next speaker is Gregory Burkeen. Gregory has pursued graduate studies at the San Diego State University at the Cell and Molecular Biology Department since 2018. And he's working as a PhD student in Dr. Anca Segall's and Forrest Rohwer's labs since 2020. His current studies focus on phage therapy for cystic fibrosis patients enrolled in clinical trials at UC San Diego. He assesses the efficacy of phage therapy via metagenomics and bacterial culture. He is also working on the production of new theoretical and in vitro models to study investigational therapies for CF lung disease as part of his doctoral dissertation. We'd like to welcome Greg Burkeen for his presentation, which is entitled New Models of CF Lung Disease.
Hey there, good morning. I'm Greg Burkeen, and I'm a PhD candidate in the Rohwer lab, and I also work for Dr. Anca Segall. And today I'm going to be talking to you about two new models of CF lung disease that our lab is using to help us make some new hypothetical treatment options, as well as to test them. Okay. And really quickly, I'm going to walk you through the general arc or trajectory of today's talk. I'm going to spend a little time talking about Trikafta and how this has made certain things possible for the research community to sort of be more helpful to physicians by doing some personalized medicine tasks. I'm going to introduce a new sort of conceptual model of cystic fibrosis lung disease, and we're calling it the sort of 4 guilds model of the CF lung. And so we're breaking the microbes down into guilds. Then I'm going to talk about a new in vitro model of cystic fibrosis lung disease that I have made, where we're taking artificial sputum media and solidifying it into these 3D bacterial culture spheres that allow us to grow aerobic, microaerobic, and anaerobic bacteria all in the same feed. And this is what they end up looking like in reality, just sort of goobers in a jar. And then I'm going to talk about how I'm using these models to help test some of these personalized treatments that are on the horizon for patients with cystic fibrosis. Okay. And as the title of the slide would suggest, I believe that Trikafta has made personalized medicine, or specifically the research community, being involved in personalized medicine possible. And I think that's mostly just a function of the math. All right, so there's around 90,000 CF patients in the world today, anywhere from 70 to 100,000. We have 30,000 here in the US. But Trikafta, the CFTR modulators, can restore function to about 90% of those mutations. And so if you go from a population of 90,000 to 9,000, and we have a population of about 3,000 cystic fibrosis labs in the world, and that's based on the number of authors with unique surnames who are publishing on cystic fibrosis in the last 4 years. That's roughly about 3 patients per lab. And of course, that's they're not all distributed evenly or next to each other all of the time. But that shift in an order of magnitude is what means that research groups can start being involved in helping physicians by providing metagenomic information and finding phages or constructing Tailocin, which is what I do. And so I'm going to talk about models of cystic fibrosis today. And those come in 3 different flavors. We have theoretical or conceptual models, and those are things that we use to help us think about the disease, how it progresses, and what we can do to try and change the dynamics in a positive way. And so here's a caption from one of Dr. Rohwer's papers with an illustration of the climax and attack model, which is the sort of conceptual model that we've been working off of for about the past 10 years. Where you have a resident population of anaerobic bacteria surrounded by another resident bacteria like Pseudomonas aeruginosa that makes that anaerobic space for them. And we call that the climax community because they are resident. And attack communities like Streptococcus, Staphylococcus, and eukaryotic viruses would be our attack community, which can come in and cause acute inflammation and acute exacerbations of the patient's lungs. One of the other flavors of model are in vitro models. So test tubes, petri dishes, or in this case, this is the Winogradsky column method. So these are glass capillary tubes, same, mostly a test tube. And then we have animal models. And I'm not going to talk much about animal models today. And first, I'm going to actually walk us through the new conceptual model that we have built. Okay. So there's a group of scientists that convenes in Telluride, Colorado at the Telluride Science Research Conference every 2 years. And this is a consensus of those scientists. We've built a model or a conceptual model of cystic fibrosis lung disease and how it progresses by breaking the microbes into functional guilds based on their what they eat and how they interact with the patient's lungs. And so the way we would actually do this is we would take a sputum metagenome. So this is a DNA extraction from a patient's sputum that allows us to run all of those pieces of DNA through a taxonomic assignment software, where we can then analyze it to see relative abundance, like what is the most abundant microbe. And we can sort of see something like this might pop up, that we might have maybe 29% Candida albicans, 10% Pseudomonas aeruginosa reads, 8% Veillonella parvula, an anaerobe, and 7% DNA from an adenovirus, which is a lytic upper respiratory virus. Then we would assemble those genomes. So go through a process of stitching all the pieces back together so that they look like a proper genome so that we can annotate those. So figure out what genes are actually written onto that string of genetic code. And we use those lists of genes present in a given microbe to sort of bin these microbes into their functional categories. And you can see here they sort of pop out to the sort of legs of this page here. And so we have 4 groups, the first of which are the nihilists. And so when you think of a nihilist pathogen, these are the ones that would burn it all down because they really don't care about the longevity of the host, or they are not well adapted to being in a host and therefore, or this host, so they cause an outsized exacerbation. This happens in normal people too. You can think of SARS-CoV-2 or strep pyogenes. The first CF specific group are brewers. And these are any microbe, eukaryotic or bacterial, that can ferment mucin and turn it into something that another microbe can eat. And then we have drunkers, of course. If you have a brewer, you're going to have a drunker. And these are the bacteria that live off of those fermentation products produced by the brewers. And these are things you would think of like Pseudomonas, a Chromobacter, Burkholderia. They're biofilm building gram-negative bacteria. And then you have the putrefying guild. And these are bacteria that have anaerobic metabolisms that produce putrid byproducts, things like putrescine, spermine, spermidine, especially putrescine. And so a couple of those would be things like Veillonella or Prevotella, which are I see pretty commonly in CF sputum. And so instead of illustrating this with a sort of PowerPoint type caption, like the previous iteration, my boss, Dr. Rohwer, has commissioned a series of paintings to illustrate this by a longtime friend of the lab and illustrator of the lab, Ben Darby. And I'm going to go through each one of these panels right now. And so the first panel is something that allows us to contrast with a healthy airway. And what you can see is that you have mucus flowing freely across the epithelial cells. And they will carry the debris, the microbes, out of the lungs along the surface of those epithelial cells, because the cilia can just beat and move it away. And because this mucin is moving so quickly, microbes are not actually able to become resident or to colonize the area permanently. But the second panel is sort of a diseased lung, but a diseased lung with maybe some colonization of bacteria, but mostly the problem is a nihilus. And in this case, nihilus are not a CF-specific guild. Anyone can have a pulmonary exacerbation when exposed to a nihilus. And in this case, what we've had depicted here is probably what you guys can recognize is SARS-CoV-2 or COVID-19 or the coronavirus. And so you can think of nihilus as being pathogens that are a sort of lone wolf. They can cause a lot of problems all by themselves, and they usually have an intracellular lifestyle, a lytic lifestyle, where they produce cytotoxic proteins. And the cytotoxic proteins, I'm thinking things like a hemolysin from Strep pyogenes, or Vibrio toxin from Vibrio cholerae, or Zonula occludens toxin, or a Shiga toxin, all of which can be highly damaging to human epithelial cells. And then we come to the first CF-specific depiction here. And what you can see is a sort of young CFer-looking lung. So you have colonization, but in this case, it's mostly dominated by the guild of pathogens called the brewers, with a sort of slight colonization from the drunkers. And so you can think of the brewers as either bacteria or eukaryotes. And so the eukaryotes could be things like Candida or maybe Aspergillus, which can cleave the sugars off of the mucin molecules themselves outside of their cells and make them available to other microbes, or things like Staphylococcus and Streptococcus, which can break down those mucins and turn them into lactate, acetate, propionate, some short-chain fatty acids that will feed the drunkards. And these drunkards are kind of the pathogens that get the most attention in the CF microbial world, things like Pseudomonas, Heterobacter, Burkholderia. And these are the ones that are sort of building biofilms and allowing our last guild to actually persist in the lungs. And so in this depiction, what you can see is that we have Staphylococcus aureus, that's the yellow here. Streptococcus are these purple microbes here. A little bit of a lag. And then Candida are these sort of green microbes depicted here. Also, Candida, I guess you could also think of as thrush, I think is a common name. And then you have Pseudomonas, and there are those blue bras there. And then we have a further progression of a sort of young CF lung towards a putrefied sputum coagulation, which is causing inflammation and cell death. So in this case, you have all three CF-specific guilds. You have the brewers, that's your Staphylococcus, your Candida, and your Streptococcus here, which are feeding the trunkers here, the blue microbes, that are drawing down enough oxygen that underneath them, you're starting to see the proliferation of putrefiers. And putrefiers are bacteria that produce toxic polyamines, like putrescine. And these toxic polyamines, though they are naturally present, all cells produce them. When you have a sort of high dwell time, high microbial load, you can reach toxic levels. Or you can just cause a lot more inflammation and neutrophil infiltration, causing more scarring. And the causative agent that we've decided to depict here is Veillonella parvula. You can see it here depicted, and the putrescine is actually being sort of represented by this sort of dead fish because it makes a sort of dead fish smell. Okay, I'm going to talk pretty briefly about animal models. And that is because in terms of animal models, we have a lot of them. They vary from zebrafish to sheep. But they vary in their ability to recapitulate CF lung disease. And this comes from a paper from the American Journal of Pathology in 2021, but it lays out that there is a sort of clear divide between animals that are good for longitudinal, long-term, and large statistically powered studies like rats and mice that are not good at translating to humans. And then when we have larger animals that are better at replicating the disease, are not good for longitudinal or being able to do a long-term study. Pigs specifically, they're the best model, but the piglets don't live very long under CF lung disease conditions. And it's hard to keep enough of the porcine models in a lab setting to do a large statistically powered study. Which is why, specifically me, I'm working on some really pretty elaborate in vitro models that can help us overcome, maybe try to reach into that longitudinal study and large statistical study group with in vitro models. And so for those of you who are not familiar with our lab's Winogradsky-based CF sputum capillary system, this is our sort of old in vitro model of cystic fibrosis, where we take an artificial sputum media that we think is similar to CF sputum, and we add indicators to it that allow us to see features of the chemistry going on inside of the tubes. We inoculate it either with sputum directly from a patient just a small bit of sputum mixed with a lot of media. Or we use a pure culture of bacteria. And then we fill those capillary tubes and put them in the incubator. And then we can observe the indicators to see what's going on inside of those capillary tubes. And we can also see gas production. And an example of this from work that Robbie Quinn and Jan Wei did was, if you take Pseudomonas aeruginosa and you load it into this artificial sputum media, and you monitor pH, the redox levels, plugging, and gas production, you can see that pH, purple means neutral. Redox, purple also means high redox. This is Comassi blue, so you can see protein aggregation. And what you see is that Pseudomonas by itself is not capable of causing these shifts in pH and redox and causing this plugging and gas production. But there is something in sputum at exacerbation that's doing that. But there are three problems with this model if we're going to try and use it to sort of fill the gap that animals have left. It's not longitudinal, it only lasts for 48 hours. The nutrient profile is actually incorrect. It's not actually representative of CF sputum. And this one is highly anaerobic, and that's due to the physical dimensions. It's about 90% anaerobic. And so the first thing I tried to tackle was, how close are we in terms of our artificial sputum recipe to the nutrient profile of real sputum? And so what you can see here on the y-axis is milligrams per mil. And I've broken that down into carbohydrates, protein, lipids, and DNA. And this data comes from a paper published by Chance and Mahandy in 2020. And what you can see is that the values of these of real CF sputum are really quite high in comparison. It's sort of a mountain in terms of nutrients, whereas we have a valley or a dearth of nutrients in our artificial sputum media. And just for fun and a quick reference conceptually, if you compare cystic fibrosis sputum to food that we would normally think of as being high protein and high fat, like salmon, it's actually nowhere near. It's just like a little baby mountain here, whereas actually CF sputum is more than twice as nutrient dense. And the closest food that comes to it, again, this is just kind of for fun, would actually be Parmesan cheese. It is much higher in fat and much higher in protein per weight than, or per volume, excuse me, than things like salmon or our old artificial sputum media. So I've reformulated the artificial sputum media so now that it contains the exact same nutrient profile of real CF sputum. Okay, and one of the other issues that model had was that it is too anaerobic. And I went through a bit of a process to try and determine what is the right amount of anaerobic. And as the title would suggest, the right amount is right around 40% anaerobic, but I'm going to walk you through the process of how we got there. And so you can imagine an airway of interest. This is a depiction of an airway in the 5th generation of the lungs, which would be 5 millimeters by 10 millimeters. And if you treat it like a cylinder, you can calculate the volume. But in order to figure out how much of that is anaerobic, I need to have some actual data about what microbialized sputum should look like in terms of how far oxygen can penetrate. And so I made some of these WinCF beads, and I used an oxygen probe to actually measure how far oxygen can penetrate. once it is thoroughly microbialized. And so there was 3.3 times 10 to the 10, so a lot of microbes, highly packed, highly microbialized artificial sputum. And so it could only penetrate about 2.25 millimeters down before it became essentially anaerobic. And if I can extrapolate that to an airway that is plugged up to a surface and air is available here, then what you could see is that I have a cylinder within a cylinder, which allows me to calculate the percent of anaerobic volume. And that would look something like 44%. And there's a small space, which is not entirely anaerobic, it's microaerobic, or should be, because a small amount of oxygen will actually make it through the epithelial cells from the bloodstream on the other side. And so this allows me to make a WinCF bead, one of these sort of mucus globs, that has the exact same proportions in terms of anaerobic to aerobic volume. And that would just take a 19 millimeter bead. And the way that I do this in real life is that I take that artificial sputum media and I add a solidifying agent. The solidifying agent that I use is sodium alginate. This is actually a pretty common ingredient in food. It can be extracted from seaweed, but it's also, if you've heard of mucoid pseudomonas, it's what makes mucoid pseudomonas mucoid. It's highly present in cystic fibrosis sputum. But what's great about it, it's a thickening agent. It makes things more viscous until you add calcium chloride. And if you add calcium chloride, this stuff actually becomes a solid and will retain its shape. And so what I do is I pour a mold made out of agar impregnated with calcium chloride that allows me to make my model into whatever shape or size I would like. And then I can move those from their mold into a petri dish, which allows me to conduct experiments on them and see how the microbes grow inside of them and see if we can try and keep them from growing. Okay. So we have several emerging treatment options. The first one I'm going to talk about today is phage therapy. I know there's a lot of interest in phage therapy because the phage doesn't care about antimicrobial resistance. A phage, if it's a lytic phage or if it's a phage that decides to be lytic, then whether or not your microbe is highly resistant or pan-resistant to known small molecule antibiotics, then it can still kill them. Though from my perspective as a microbial ecologist, what I would like to study and I would like to understand is not only how I can use a phage to kill one microbe in a cystic fibrosis lung, but actually how can I use this to affect the microbial ecology in a way that might actually help the patient. Because in terms of ecology, if you empty a niche, but you haven't removed the niche, it will get filled by something else. And so I have done an experiment where I made these beads, and I have a control group and I have a phage treatment group. And what's good about these beads is it allows me to test things that you could never test in a patient. And so I inoculated this WinCF bead with sputum from a patient. And and I incubated them at 37 degrees C. And then I would sacrifice a number of beads, 3 beads every day, or at time points in the study, to see how many colony forming units were growing in those beads at that time. And then I treated them, instead of with a cocktail of maybe 1 or 2 phages against a single pathogen that we know lives in this sputum, I tried to find a phage for every pathogen that we know is in this sputum, because I had a metagenome of this sputum before I started this experiment. And so this included every phage in our library. And this also included filter sterilized agnostic phage sources, meaning things like sewage, river water, lake water, ocean water, sometimes food, compost, and things like that, where we find a lot of lytic phages. And so this way, there should have been a phage for most of the bacteria that were present in this patient's sputum metagenome. And then I treated that mucus plug model with the phage cocktail. And that's what is here in orange on this day. And what I saw was that there was essentially statistically no difference in the phage or no phage control, but that sometimes total microbial load it is very erratic, which is also something that we see in cystic fibrosis sputum samples. So this, again, this doesn't have a lot of bearing on how phage therapy is going to do in the clinic for cystic fibrosis patients. This is specifically just me showing you how I can use this new model to test things that might or might not work, which can help us make better treatments in the future, and help us understand what's going on in cystic fibrosis sputum. And that this system is also erratic in terms of the microbial load from day to day, is actually a good sign in my mind, because it means I'm actually recapitulating what we see in a real patient. And in terms of being able to accomplish a longitudinal study that remains dynamic in an in vitro system, this is what gives me hope for this system, as well as my next topic, which is so far a hypothetical treatment option. This is not in the clinic yet, but there is good news on this front, and I'm here to tell you about that today. And so tailocin therapy, you can think of a tailocin as a phage without a head. So essentially, this is the same as that phage before, but it doesn't have a capsid that contains DNA, which means that it cannot reproduce, and it will bind to a target, and it will just kill it. Okay, and so our lab has a relationship. We have a material transfer agreement from Pylum Biosciences, and they have allowed us to use their avidicin construct, specifically to try and retool that tailocin, which normally comes from Pseudomonas, to kill Stenotrophomonas multifilia. And here's a picture of what that looks like. It's a protein structure that has essentially a spring-loaded harpoon that you can sort of see here. This holds energy, which will push the harpoon through the bacterial cell membrane and the cell wall. but it has to bind very specifically to one type of bacteria, and that's facilitated by whatever's on the end of these tail fibers and at the tip of this tail spike here. So it has to actually sit down on a bacterial surface and bind before it will release its payload. And so my goal since I started this project in the late summer of 2020 has been to take that construct and to stitch a tail spike, which comes from a phage, onto this avidicin construct, a phage that's specific to killing Stenotrophomonas multifolium. Okay, and the reason that we think that this can be safer than a phage in general is that since they cannot reproduce, you can actually control the dose. A phage will amplify inside of the host, which can be a good thing, or it could also be a bad thing in case it leads to the release of too much endotoxin all at once. And so in this case, you can moderate the dose. Because it has no genome, it also cannot conduct horizontal gene transfer, meaning it cannot pass a gene for one of those toxins I was talking about before from one microbe to another. And it also can't pass antibiotic resistance genes from one to another, which phages are quite notorious for doing. And it can only kill. A phage has a choice, or a lot of them have a choice. Some are obligate lytic, but some phages have the option to be temperate, meaning that they can choose to kill or not to kill. This tailocin can only kill. Okay, and as of Monday morning of this past week, an anti-Stenotrophomonas tailocin was born. I have actually managed to build this thing, though it started in the summer of 2020. And so in order to get there, it's actually kind of a long process, and I'm going to walk you through that before I show you the data. And so the very first thing you have to do is you have to find a phage that can actually kill Stenotrophomonas multifolium. And not only do you have to find one that can kill it, but you have to find one where you can recognize the tail spike. Because you're going to take that tail spike and you are going to string it onto the end of that tail fiber so that it will sit down onto a new bacteria and kill that bacteria. And you have to amplify that up using PCR, and then you have to assemble it into a plasmid that will create a fusion protein between this tail fiber and your new tail spike that allows you to change the specificity of this bacteria killing machine, this headless phage. And it's important that that plasmid be inducible so that you can produce a lot of it all at once, meaning you can add something to the bacteria and it will produce a ton of this protein for you. You need to transform that or put that plasmid, that DNA, into a bacterial host strain that has the rest of the structural genes of that tailocin construct. And then you need to run the induction, and that'll produce a ton of tailocin for you. And then you take that supernatant from that induction event, and you can add that to your target strain of bacteria. And then you can measure the amount of bacteria that were killed compared to a negative control. And that's what I did Monday morning. And here is the actual data to support that I have made a functional Stenotrophomonas multifilia killing tailocin. What's here on the y-axis, these are the number of colonies on the plate that I counted. So a colony means that there was a bacteria present. And what I did was I incubated the Stenotrophomonas multifilia with the tailocins for 30 minutes, and then I diluted it to 10 to the negative 4, or 10,000 times. And then I took a 100 microliter sample of that dilution, and I spread it on a plate of nutrient agar. And wherever there was a bacteria, I would see a colony. And the negative control here in blue gave me about 66 million colony forming units per mil. But the Tailocin treatment gave me just 5 million, or 50 million colony forming units per mil, or 5 times 10 to the 7. Which means that that induction event was able to produce about 10 to 15 million Tailocin killing units, which is pretty exciting, and is actually kind of a big step forward in this specific sort of research field, or well, in this research direction. And I want to acknowledge everyone in the Rohwer lab and the Segall lab. I have 2 bosses, Dr. Forrest Rohwer and Dr. Anca Segall. I want to make a special shout out to Cole Souza and Hannah Jalazadeh, who are both working with me pretty closely and did a lot of the work in producing that Tailocin. And of course, my funder is the Cystic Fibrosis Research Institute. And thank you very much. And I am open for questions, and I will navigate back to whichever slide anybody has a question about.
Greg, thank you so much for your incredible presentation. So I think we have about 7 minutes left for some Q&As. Let me just go through here. So I'm going to read the comments that are in the chat right now. So Devin Wakefield is commenting, just now my doctor told me that prioritizing treating the brewers doesn't seem to have a big impact in CF care. Is that right? Is there not much clinical evidence to support this model? Or does this mean that we just don't have great antibiotics treatments for fighting brewers? Or will the other colonizing bacteria still be able to grow in the lungs even if the brewers are killed off?
So the brewers, so it depends on which brewer you're talking about. So the brewer Candida albicans, or possibly Aspergillus, these are eukaryotes. And as I understand, in speaking with one of my committee members, Dr. Conrad at UCSD, is that there aren't great treatment options for things like Candida, because they're also a eukaryote and we're a eukaryote. But in terms of trying to treat the brewers, things like Staphylococcus and Streptococcus, I would say that it probably, if you're looking to try and reduce the amount of brewers, it may be that it really depends on which brewer you're talking about. Staphylococcus and Streptococcus, you could probably have a better impact on reducing the number of those using something like hyperbaric oxygen therapy, or just hyperoxic conditions, because these are mostly microaerobic or back anaerobe bacteria. They like the oxygen less. That would be what I would think.
All right. So then we have a question from Scooter Thornton just now. Is there a process to determine how many Tailocins will be required to eliminate an infection in a patient?
That is the next experiment on my docket. I will be using our new in vitro model to test exactly that. So that'll be just this upcoming week, I'll start work on that.
So then we have another question from Devin Wakefield. If oxygen has a hard time penetrating in mucus plugs, does that also imply that inhaled antibiotics have a hard time penetrating as well? What can patients do to combat this?
The rate of diffusion through that microbialized mucus is not necessarily hindered by the microbes or the density itself. It's more dependent on the number of microbes and their metabolism drawing down the oxygen. And so if you have a small molecule, an aqueous small molecule, it should be able to make it through or at least some distance into the substrate, though the issue I think you would run into is more to do with anaerobic cells and facultative anaerobic bacteria that can be persisters, meaning that they, when their metabolism shuts down and they start doing low efficiency anaerobic metabolisms, this allows them to sort of persist past the anaerobic or the antibiotic treatment. This is something I've seen with microbial strains again and again, is that the inhibitory or bactericidal concentration is much higher if you are growing them anaerobically.
Okay, thank you. So we have a next question from Naren Kumar. How well do tailocins penetrate the plug?
I wouldn't know yet. That's something that I'm going to have to test. And again, they didn't exist until, these tailocins did not exist until Monday morning.
Wow, thank you for this wet ink data. So then we have one question from Martina Gensch. You mentioned that you are rebuilding mucus in vitro. Are you taking into account the different mucin concentrations in CF versus non-CF mucus?
Yes, the model is the recipe is specifically tailored to cystic fibrosis patient sputum, and that's determined empirically in that paper by Chance and Mahoney, where they took 468 different sputum samples and then told us what the nutrient values were and what the constituents were. And so it's all based on CF sputum, not normal sputum.
Okay, Gregory, so we don't have any more questions coming in. I think we have 1 minute remaining. And let me thank you from the bottom of my heart for your incredible presentation. That was very artsy, very scientific, and we welcome you to the CFRI research community. We're so grateful to have you. Yeah, thank you again for this incredible presentation. And I would like now to take a 35-minute break. So feel free to explore the exhibitor hall or check out the activities in the lounge. And we will reconvene here at 12:45 Pacific, 3:45 Eastern time for the final research presentation. Thank you all and see you in 35 minutes. Bye-bye.